Hello everyone, welcome to the channel Microbial World. Today we will be discussing about the bacterium Bacillus anthracis, its morphology, transmission, virulence factors, pathogenesis, clinical manifestations, laboratory diagnosis, treatment and prophylaxis will be discussed in detail. Bacillus anthracis is the causative agent of a serious zoonotic disease called anthrax. Zoonotic disease are diseases that are transmitted from animals to humans, okay? And they can spread by direct contact with infected animals. Anthrax is a disease of wild and domestic herbivorous mammals. It is one of the most common agent of bioterrorism, which means Bacillus anthracis is used to cause harm to humans, animals and plants by releasing this biological agent at a specific place. It was used in Verdol's anthrax outbreak in 1979 and US postal system attack in 2001. Bacillus anthracis was first isolated from infected domestic animals by Kohn in 1872. Morphology. They are gram-positive rods arranged in chains. It is an aerobe and a facultative anaerobe. The cells are of size ranging from 1 to 1.2 micromuem in width and 3 to 5 micromuem in length that occur either singly or in pairs. The outer covering of Bacillus anthracis is made up of capsule, which is made of polyglutamate, extensive peptidoglycan layer, lipoteichoic acids, and crystalline cell surface protein S layer. They are endospore forming. They form non-bulging spores that are central or subterminal, elliptical or oval in shape. They are non-motile. Transmission. Humans and animals acquire anthrax infection by ingestion, inhalation, or traumatic inoculation of spores of Bacillus anthracis. Spores are dormant forms that are found in the soil or in contaminated animal products like hair or animal hides, that is the skin of the animals. Okay. Virulence factors. Pathogenesis of anthrax is due to two virulence factors, that is anthrax toxin encoded in plasmid PXO1 and capsule encoded in plasmid PXO2. Capsule is polypeptide in nature. It is composed of polymer of D-glutamic acid. So how does the capsule aid in virulence? That is by inhibiting phagocytosis. Now, capsules are not formed under ordinary conditions of culture. Capsule formation occurs only if the media contains added bicarbonate or if incubated under 10 to 25% carbon dioxide, or if grown in media containing serum, albumin, charcoal or starch. Capsule formation will occur in the absence of carbon dioxide. Okay. Anthrax toxin. It is a tripartite exotoxin, which means it is a complex of 3 fractions. Now these 3 fractions, they are not toxic individually, okay, but the whole complex produces local edema and generalized shock. The 3 fractions are protective antigen factor, edema factor and lethal factor. Protective antigen factor is a receptor binding protein that binds to the host cell receptors, and it facilitates the entry of other fragments, that is edema factor and lethal factor. Edema factor is the active fragment. It acts as adenylate cyclase enzyme. Okay, so what is the function of adenylate cyclase? It will convert ATP to cyclic AMP, right? So, edema factor will increase the host cell cyclic AMP levels. It is responsible for edema, which means swelling of the tissues, okay, and other manifestations seen in anthrax. Lethal factor is a metalloprotease that is responsible for cell death, but the mechanism of action is not known. Pathogenesis. The mechanism of action of toxin involves a series of steps. So we said that the protective antigen factor is a receptor binding protein, right? So the first step is protective antigen binding to specific receptors on the surface of target cells such as macrophages and endothelial cells. Next step is endocytosis. Once the protective antigen is bound to the receptor, it is cleaved by cellular proteases. This results in the formation of protective antigen heptamer. This heptamer binds to additional protective antigen molecules, thus forming a pre-pore complex on the cell surface. Next is translocation. The pre-pore complex will undergo conformational changes, allowing it to insert into the cell membrane and form a pore. This pore will serve as a channel for the translocation of edema factor and lethal factor into the cytoplasm of the target cell. So we know that the edema factor is an adenylate cyclase enzyme, right? So it will convert ATP to cyclic AMP within the cell. The increased levels of cyclic AMP will disrupt cellular signaling pathways, thus leading to increased fluid secretion, tissue swelling, that is edema, and impaired immune cell function. Lethal factor is a zinc-dependent metalloprotease that will cleave and inactivate key proteins involved in cell signaling pathways, including those involved in inflammation and immune response. Thus, it will lead to cell death, tissue damage, and systemic effects. Clinical manifestations. Cutaneous anthrax. So, cutaneous anthrax affects our skin. Okay. The infection, it results from direct inoculation of spores via small cuts or abrasions. Lesions called eschars. Eschars on skin, they are very dry, dark scab. Okay. These lesions, they are found on exposed regions. For example, hands, arms, neck, wrist and face of the body, accompanied by edema. And this disease, it was also common in dock workers who carried hides and skins of the animals on their bare backs. So this disease, it was also known as hide porter's disease. Incubation period ranges from few hours to 3 weeks, and most often it is 2 to 6 days. Pulmonary anthrax. So we know that the word pulmonary refers to anything related to the lungs or the respiratory system, right? So pulmonary anthrax, it is also called wool sorter's disease because it was common in workers in wool factories due to inhalation of spores from the infected wool. Symptoms include fever or chills, sweat, fatigue or malaise, non-productive cough, dyspnea, which means shortness of breath, changes in mental state including confusion, and nausea or vomiting. It can also cause hemorrhagic pneumonia with high fatality rate. Hemorrhagic meningitis can occur as a complication. Incubation period ranges from 4 to 6 days. Oropharyngeal or gastrointestinal anthrax. Oropharyngeal refers to part of the throat that includes both the oral cavity, that is our mouth, and the pharynx. Gastrointestinal system, it is our digestive system, right? And in case of oropharyngeal anthrax, lesions are seen in buccal cavity, tongue, tonsils or posterior pharyngeal wall. And in case of gastrointestinal anthrax, lesions are mostly seen in ileum and cecum. Symptoms include sore throat, dysphagia. Dysphagia means it is difficulty in swallowing, okay? Vomiting, mild diarrhea and fever. Violent enteritis with bloody diarrhea can occur with high fatality. Enteritis, it is the inflammation of small intestine. Incubation period ranges from 3 to 7 days. Anthrax meningitis or meningoencephalitis. This is a fatal infection which occurs when organism enters the central nervous system via the bloodstream. Injectional anthrax. This is a new form of anthrax that is found in injectional drug users. Okay, the symptoms, they are similar to cutaneous anthrax, but there can be infection deep under the skin or in the muscle where the drug was injected. And this disease, it will spread throughout the body fastly and it is very hard to recognize and treat. Laboratory diagnosis. Specimen. Fluid or pus is taken from lesion in cutaneous anthrax. Pleural fluid, blood or cerebral spinal fluid is collected in inhalational anthrax or associated sepsis, and stool is taken in case of gastrointestinal anthrax. Microscopy. On gram staining, gram-positive bacilli is observed. McFadden's reaction. Polypeptide capsule can be demonstrated by staining with polychrome methylene blue stain for 30 seconds. So the capsule will appear as amorphous purple material surrounding blue bacilli. Okay, so this McFadden's reaction is used for presumptive diagnosis of anthrax in animals. Okay. Direct immunofluorescence test. It is used to detect capsular and cell wall polypeptide antigens using fluorescent tagged monoclonal antibodies. Ascoli's thermoprecipitation test, which is also called ring precipitation test. So in this test, tissue samples are taken from suspected animals. It is grounded and suspended in saline. Then it is boiled for 5 minutes and filtered through a filter paper or muslin cloth, after which it is allowed to cool. Then anthrax antiserum is taken in a narrow capillary tube. The antigenic extract is layered over the anthrax antiserum. A positive test will show a ring of precipitate at the junction of two liquids within 5 minutes. Culture. Bacillus anthracis is aerobic, non-fastidious. It grows in ordinary media and it has a temperature range of 12 to 45 degrees Celsius of growth. On blood agar, Bacillus anthracis produces dry, wrinkled, non-hemolytic colonies with frosted glass appearance. And sometimes the edge of the colony is composed of long interlacing chains of bacilli. It resembles the locks of matted hair. Okay, and this is called medusa head appearance. On gelatin stab culture, inverted fir tree appearance is seen. This is because of slow liquefaction of gelatin commencing from the top. Okay, so this is somewhat how the inverted fir tree appearance in gelatin stab culture looks like. Okay, and as we said, it is because of the liquefaction of gelatin starting from the top. On solid media with penicillin, the colonies will have a string of pearl appearance look. So why does this show this appearance? It is because under the action of penicillin, the cell wall becomes weak and the cells will become larger and spherical and it will occur in chains. Selective medium is plet medium. It consists of polymyxin, lysozyme, EDTA and thallus acetate added in heart infusion agar. This medium is used to isolate Bacillus anthracis from mixtures of other spore-bearing bacilli. Biochemical reaction. They are indole, Vogt-Prosker and citrate positive. Methyl red, oxidase and urease negative. Glucose, maltose and sucrose are fermented, producing acid but no gas. Nitrates are reduced to nitrites. Catalase and coagulase is formed. Animal inoculation. On subcutaneous inoculation of culture into guinea pigs, the animal dies in 24 to 72 hours. It shows local gelatinous hemorrhagic edema at the site of inoculation, extensive subcutaneous congestion, and an enlarged dark red spleen. The blood is dark red and it coagulates less firmly. Serology. Antibodies appear in convalescent sera and it can be detected by ELISA or indirect hemagglutination methods. Molecular diagnosis. Polymerase chain reaction with specific primers can be used for confirmation. Molecular typing. MLVA, multiple locus variable number tandem repeat analysis, or AFLP, amplified fragment length polymorphism can be used. Treatment. The bacteria is sensitive to many antibiotics such as ciprofloxacin, erythromycin, penicillin, vancomycin, amoxicillin and doxycycline. Antitoxin therapy, that is antibody against protective antigen, is used with antibiotics. Okay, human anthrax immunoglobulin, anthracil and monoclonal antibodies to protective antigen, that is obiltoxaximab and raxibacumab is being used. Prophylaxis. So the disease can be avoided by maintaining proper hygiene and protection during the handling of infected animals. Anthrax vaccine adsorbed vaccine, AVA Biothrax. This is approved by FDA and it is for human use obtained from the culture of Bacillus anthracis having protective antigen. Okay, so this is all about Bacillus anthracis. Hoping that the topic was clear for everyone. Thank you.